Hello everyone, how are you all doing? Good evening everybody. Good evening Jasmine, good to see you here. All right, so yes, who else are there? Can we have some quick highs and around? How's everyone doing today? So in the last session, we have talked about gene mapping. I told you that this, anyways, it's just one subtopic of entire unit 8, but so many questions come from this segment that I had to break it down. So in the last session, we talked about linkage maps. In this session, we will further talk about gene mapping only, but we will mostly take the questions which are related to molecular markers. Have you people seen this kind of question where they give you a pattern, the band pattern in agarose gel electrophoresis, or how the band pattern comes in southern blotting? So they will give the band pattern. So that is basically showing the presence or absence of various markers or how the markers are formed there. And you have to analyze it to answer the questions. I'm also doing good, Jasmine. Thank you. Good evening, Shrivani. All right, so let's start. Okay, without any further delay, let's start the question discussion here. Now, before that, I would like to give you a brief idea. So in the last class, we have talked about linkage mapping. Okay, so I told you this entire mapping thing is just like how we do the the Google mapping, the geographical mapping, where we map places in the G in the city, right? We use landmarks, we place them in the different locations of the city so that we know where exactly each thing is, and also it becomes easy for us to navigate from one place to another. The entire idea is similar here for gene mapping also, but in linkage mapping, it's not the absolute distance. Okay, it's like when we say distance between two things in terms of minutes, like this much time you will take to travel from one place to another. So time is definitely not a unit of distance. Okay, but still we say that, and that gives us an idea how far or how close a place is. That is how linkage mapping is. So in linkage maps, we get the distance in terms of recombination frequency. How frequently two genes will recombine, that gives us the recombination frequency, and this gives us the genetic distance between the genes. So recombination frequency, if it is high, means the genes are present far apart. If the recombination frequency is low, means the genes are present close. Okay. Physical mapping is not like that. When we talk about physical mapping, yeah, good evening, Taban. Okay, so when we talk about physical mapping is about the absolute distance between the genes. Okay, so physical map always give us distance between the genes in terms of kb. Okay, it gives us distance in terms of kb. All right, low base pair. And then for physical mapping, we map with the help of various molecular markers. Okay, now markers are not required to be genes. They don't have to be gene. They can be any unique sequence, any non-coding unique sequence, which can be that is what is called as a marker. Okay, so they will be basically landmarks. All right, but those landmarks, they need not to be gene. Okay, they can be any sequence which is unique to that particular location. That is what we will call that is what we call them as the molecular markers. Okay, so rflp, rapd, aflp, so these are all molecular markers. Now, with the help of molecular markers, when we do the mapping, so we get the idea that with respect to that marker, how far is our particular gene is present in the genome. Okay, now I think I have given you enough background. So let's dive in and try to solve some difficult part C questions from this segment. Are you all ready? Can I have some quick BIOs in the chat to know that you guys are ready? ठीक है, और ऐसा करती हूँ कि मैं अपना वीडियो बंद कर देती हूँ सो दैट स्ट्रीमिंग में कोई दिक्कत ना हो, ताकि हमारी स्ट्रीमिंग स्मूथ चले एंड द वॉइस रिमेन्स क्लियर. Okay, I'll just turn off the video. 1 second. Right. Okay. Now again, I would like to tell you, especially the ones who are joining the first time here, that please stay till the end of the session and we'll be discussing questions now. So whatever doubt you get in the question, whatever part you did not understand and you want to have a one-on-one session on it, so the Zoom link for the class will be provided to you at the end of the session. So please join the Zoom link and clear your doubts. Okay, so you can write down whatever doubt you are having right now when we are solving the question, that specific question, and we will see it at the end of the class. You can have a one-on-one discussion with me, and we can like you can have a better understanding of the question. Okay, and same way, just like how it happens in the previous sessions also, that some freebie links will be provided. Okay, so those links will help you, will give you some stuff. Okay, that's a surprise, but it will give you some free stuff, and that will help you with your CSIR preparation. Okay, so let's start. Okay, so what do we have here? So a figure below represents a profile of DNA markers in two parents, P1 and P2, progeny F1 from a cross between P1 and P2, and that of the gametes produced from F1. 8 different patterns, DH1 to DH8, were observed in each in case of gametes. Numbers below DH1 to DH8 indicate the number of individuals observed in each case. Okay, so basically what it is saying is P1 and P2 are two parents. We are crossing them. We have got F1. Okay, now this F1 is producing gametes. Okay, so we have got 8 different pattern of gametes. So what pattern we are looking here? We are not looking for genotype. We are not looking for the phenotype. Definitely gamete cannot have a phenotype. But the gametes here, we are looking for the DNA marker, the pattern of DNA markers in these gametes. Ankita, from the setting of your YouTube, you can choose the video quality. Okay, it's coming through the screen also. Please follow that and you will get the better quality video. Okay. Yes, so 8 different patterns, 8 different patterns of the DNA marker that we are analyzing here is we have got in 8 gametes. Okay, so these gametes are marked from here. So basically DH1 to DH8 are the gametes of F1. Okay, and this numbers in the bracket which is shown below, so that is the number of each gamete. Okay, numbers below DH1 to DH8 indicates the number of individuals observed in each case. Okay, now based on the above observation, the following statements are made. Markers B and F are likely to be allelic in nature. Okay. So, and B statement says that markers C and D are linked in trans with a map distance of 24 centimorgan. And C says that marker B assorts independently for marker C. Okay. So we will check each of this statement one by one. Now checking the first statement here, marker B and F are likely to be allelic. Okay. Now, first of all, what is an allele? Okay, going to the very basic discussion of genetics. So allele is nothing but an alternate form of a gene, right? So if it is allelic in nature, now what does that mean? That both the alleles will occupy the same gene locus. Correct? Alleles. What are alleles? Alternate form of gene. Okay. So alternate form of the same gene it is. That means they will occupy the same gene locus, right? And one more thing, what did Mendel found out about the alleles? He said that at the time of gamete formation, the alleles will segregate from each other. Remember the second law, the law of segregation. Okay. Now to analyze if B and F are allelic in nature, see, this is B and this is F. Okay. So B is coming from P1, F is coming from P2. F1 has both B and F. Okay. Now the gametes of F1, if you see, okay, so D1 to D8. Now if if one is having B, it is not having F. If one is having F, it is not having B. So do you have any gamete here which has B and F both, or B and F none? It's not there, right? Either one of them it is having, but only one it is having, not both. Correct? Okay. What does that mean? That it might be the allele of each other. That is why they are segregating. They will assort, they will segregate into different gametes. Okay. They will never come together. It's not possible because the chromosome number will be halved here. Okay. So B and F might be allelic in nature because they are segregating at the time of gamete formation. Okay. So A statement can be correct. What about the B statement? It says that the markers C and D are linked in trans with a map distance of 24 centimorgan. Okay. Now, what is trans configuration? We have told in the last class, right? So trans configuration means where both of them are present on different chromosomes. Okay. What it is saying about C and D. Okay. C and D. Now, do you see here that C and D, they both are present on the chromosome that is coming from P1. P2 has none. Okay. What does that mean? That from P1, the chromosome that is coming, it has both C and D in it. Okay. So if they are present on the same chromosome, will we call it trans? Can I have some quick answers? If both C and D are present on the same chromosome, will we call it trans configuration? It would have been trans configuration if one is coming from one parent and other one is coming from other parent, right? Here it is both coming from the same parent. Okay, so if they are present on the same chromosome, then what is the configuration here? It is cis configuration, right? So C and D are linked in trans. That is wrong. It is present in cis. Okay. Now, if this is wrong, there is no point of checking the map distance here. Okay. Right, but you can still check the map distance. Now, how can we check the map distance? So the map distance, for that we need the recombination frequency. So for that we have to find out the recombinance. Okay, now see, this is a parental combination where both C and D are present. This is a parental combination. Okay, this is a parental combination. In fact, this one is also a parental combination, right, from P2, where none of C or D are present. Okay, This is also a parental combination. Okay. But this one, that is DH5 and DH7, DH6 is also parental, DH8 is also parental. DH5 and DH7 are the two varieties which are showing a recombinant combination, right? Where only one is present, other one is not present. Yeah, good evening, Amit. Good to see you. Okay, so what it will be? So these are the recombinants. So when we are calculating the recombination frequency, how we will get it? It will be the number of recombinants. So number of recombinants is how much? 10 and 14. So 10 plus 14 divided by total number of gametes. When you add them up, it is 100. Okay. And multiplied with 100, that will give you the recombination frequency percentage, which is the distance. So 24 centimorgan is the distance that is correct. Okay, but C and D are not linked in trans. So this statement is wrong. Third statement says that marker B assorts independently from marker C. Okay, now I'll just clear the screen here. Marker B assorts independently from marker C. So B and C. Okay. Now again for B and C, if you see, so B and C are present, are coming from P1 only. Okay. They are both absent in P2. Okay. So if B and C are getting assorted independently, then what should be the situation in independent assortment? In case of independent assortment, the recombination or the proportion of recombinants should be equal to the proportion of non-recombinants, right? Each should be 50%, isn't it? If the genes are independently assorting, recombinants and non-recombinants should be in equal proportion. Okay. Right, now let's find the recombinants here for B and C, just how we found out the recombinants for C and D. Okay, so you have to consider B and C. So here DH1 is a parental combination. Okay, DH2 is again a parental combination. So I'm just writing P here, P here. DH3 is also parental. Okay, now DH4 is recombinant. Why? Because C is present, but B is not present. DH5 is also recombinant because B is present, but C is not present. Okay. DH6 is parental, DH7 is parental, DH8 is parental. Okay. So what are the recombinants? Only 4 and 5. Okay. So recombination frequency or recombinance frequency, how much it is? 10 plus 11 divided by 100. Okay. So it is 21. So 0.21, okay, or it is 21%, but it is not 50%, okay. So that is why recombinants are far lesser than the parentals. That means B and C are not independently assorting and they are linked. Okay, so that makes our C statement also wrong, which means answer will be option 3, only A. I hope all of you understood this. So here you did not have to do many, a lot of calculation, very easy calculations only were involved, but only thing here was analyzing. Okay. So whenever this kind of questions are there for which are talking about this maps and bands are coming. So the major thing is analysis. Okay. They are completely based on the analysis. All right. Now moving on to the next question. So guys, if you have any doubt in the previous question, please write it down or join the Zoom class and we will take it up there. Yes. So in normal individuals, there are 3 MST2 restriction sites, 2 flanking the beta globin gene and 1 within the gene. If individuals affected by a disease and a single nucleotide polymorphism in beta globin gene abolishes the internal MST2 recognition site, the RFLP pattern for this locus obtained by hybridization using a probe internal to the flanking MST2 site from 3 siblings of the family is shown below. Okay, this is shown here in the table. Based on the above profile, what is the nature of the genetic disorder? Okay, so this question can be considered as a part of pedigree analysis also, because we are talking about how the genetic disorder is getting passed, okay, is getting inherited, which is something that we analyze in pedigree analysis. Okay, and the other thing is the information that we are getting on the table, which is the molecular markers. Okay, so that's why you can consider it as part of any of the concept, does not matter, we will take it up here. Okay, so it is the RFLP pattern is given. So first of all, what is mentioned in the question? So the question says that in normal individuals, the beta globin gene, okay, it has 3 MST2 restriction sites. So I will draw the pattern for normal. Okay, so in case of normal, what happens? So if this is the beta globin gene, so it has 3 MST2 restriction sites. 2 flanking the gene, means one here and one here, and the entire beta globin gene is in between. Okay, and one more site is there which is within the gene. Yes, Amit, you are correct. Okay, so we'll see how it is, right? So this is the normal genes pattern. Okay, so basically when you digest the normal normal copy of beta globin gene with MST2, so there you will get 2 fragments. Okay, so digestion with MST2 will produce 2 fragments. Okay, now what about the deceased? So in case of deceased, we have what happens? These 2 sites are present. Okay, the sites which are flanking the beta globin gene is present, but the internal site is absent. Okay, so if you digest the deceased copy with MST2, what will happen? Only 1 fragment we will get. Okay, and this 1 fragment will be a bigger fragment. Okay, so addition of these 2 fragments will be the 1 fragment that we got in the deceased alleles digestion, right? Okay, now based on the above profile, what is the nature of the genetic disorder? Now let's check the the profile that is given. Okay, so it's the siblings profile it is given. Okay, so first of all, it is saying that there's a normal son. Okay, now he has 1.35 fragment size of 1.35. One is 1.15, one is 0.2. Okay, so how come this normal individual is having 3 fragments? So if you see this 2, okay, this 2, when you add it up, it becomes 1.35, right? So what does that mean? That this son has 2 different copies. Okay, 1 copy is normal, right? And 1 copy is deceased. So this individual has a pair of homologous chromosome. One of that has the normal copy. The other one has the deceased copy. Okay, now instead of writing it in forms of chromosome, okay, so I will write it for this. Okay, so I will draw the DNA. So basically it will have this normal son has 2 DNAs. Okay, one has the mutation, and because of which it is forming this one bigger fragment of 1.35, whereas the other DNA molecule that the son has, where again a copy of this gene is present, it has the normal copy, and because of that there are 3 digestion sites are present, and that's why 2 fragments are coming from this DNA, 1 fragment is coming from that DNA. Okay, so the normal son, first of all, it's a son has 2 copies of the gene. So that definitely it means that the gene cannot be X-linked, because if it was X-linked, the males will have only 1 copy. Since the son has 2 copies, means it has to be autosomal. Okay, now in autosomal condition also, you see that the son has 1 deceased copy. Okay, this one is the deceased copy. The son has the deceased copy, but still he is phenotypically normal. Okay, what does it means? That it is the recessive allele. That's why when a normal copy is present, the deceased allele is not showing its effect. Okay, so just by analyzing this column only, this normal son only, you will get your answer that the of the genetic disorder is autosomal recessive. Okay, you can further confirm by analyzing the other two columns. So the normal daughter, okay, she has only these two fragments. So basically it means that she has two copies and both of them are normal. Okay, so from both of them it is producing the two fragments, so they will come together and form the same profile. Affected son also will have two copies, but both the copies are deceased copy. Okay, so that's why the son is having only one band of 1.35 kb. Okay, and this son is affected because the allele was the deceased allele was recessive, and now it is present in homozygous condition, so it will show the disease phenotype, and that's why this son here is affected. Okay, I hope you all understood. Now again, as you see here, only analysis we have to do. Next question. Now this is a question from deletion mapping. Okay, deletion mapping, which is a technique for doing the physical mapping in a physical mapping of genes. Okay, so the location of 5 overlapping deletions have been mapped to a Drosophila chromosome as shown below. So there are 5 deletions are showing, deletion 1 to 5. Horizontal lines in the above figure indicate the deleted regions. Okay, so wherever the horizontal line is there means that region is deleted. Okay, so basically for deletion 1 here means this much segment has got deleted. Okay, for deletion 2, so in the this chromosome, this much segment has got deleted. Okay, like that. Now recessive mutations A, B, C, D, and E are known to be located within these regions, but the order of mutations on the chromosome is not known. Okay, so we know that this segment, this entire segment of chromosome has A, B, C, D, E recessive mutations, but where exactly which one is present, we do not know. Okay, when the flies homozygous for the recessive mutations are crossed with flies homozygous for the deletion, the following results are obtained. Okay, letter M represents mutant phenotype, plus represents wild type phenotype. Okay, there can be another form of representing this table where instead of writing M as the mutant phenotype, it can be written as minus as the mutant phenotype. Okay, but ultimately it will be the similar condition. On the basis of the above data, the relative order of 5 mutant genes on the chromosome is. Okay, that is what we have to find out here. Shivani, if the video is not clear for you, please go to the settings of the YouTube and change the pixel there. Okay, I request the biotechnical team to flash that information on the screen. That will help the students to get a clear video. Please, team, if you can flash it up on the screen. Yes, the information is coming here. The steps are written. Please follow that and you can get a better quality video. Thank you. Right, now coming back to the question here. Okay, so what is the basic idea? Okay, what we have done here is we have a fly who is homozygous for the recessive mutations. Okay, crossed it with another fly who is homozygous for the deletions. Okay, deletions in different places. Okay, so the heterozygote, the F1, okay, what it will have? It will have the chromosome which is in all the recessive mutations. Okay, let's say this is the chromosome. So A, B, C, D, E. Okay, randomly I'm placing it. I don't know. Okay, to make you understand. And the other chromosome that the fly will have deletions. Okay, now suppose this fly has this deletion. Okay, the other chromosome has this deletion. Okay, so D and E are present. Okay, the dominant D, dominant E are present, dominant C is present. Okay, but A and B, the segment of A and B where A and B is supposed to be present is not here, it is deleted. Okay, so in this situation, what will happen? What will be the phenotype? Okay, so in terms of C, D and E, the phenotype will be of the dominant allele, right? Because one copy of them is present. Okay, but what about A and B? The copies of A and B are absent here. Okay, but the cell do not have any other copy. Okay, so in that situation, this recessive alleles, even though present in a single copy, will express themselves, and that is something that we call as pseudo dominance. Okay, so this recessive alleles, because its corresponding segment is absent in the other chromosome, this recessive alleles will show pseudo dominance. Okay, so what will be the phenotype? For C, D and E, it will be plus. Okay, but for A and B, it will be the mutant phenotype. All right, now this is the basic concept of doing a deletion mapping. Okay, so basically the genes which are present on the deleted segment. Okay, so what we conclude from here, so the conclusion will be that the genes which are present on the deleted segment, okay, they will show the mutant phenotype. Okay, so genes on deleted segment will show the mutant phenotype. Okay, whether you call it mutant phenotype or recessive phenotype or whatever you call, but that is what will happen. So the genes which are present on the deleted segment, they will show the mutant phenotype. The genes which are present in the non-deleted segment, they will show the wild type phenotype or the dominant phenotype. Okay, now with this, let's come back to the table here. Okay, this first table, the deletion. Okay, so what we will do is, just looking at the deletion, let's assign the phenotype. Okay, so for this part, this first section, okay, so it is, see, you see the entire chromosome has been divided into 6 sections. Okay, so for this sections, let's see. So for this section, what will happen? Deletion 1 has got deleted here. Okay, so what phenotype will it have? Mutant phenotype. So any gene which is present here will show the mutant phenotype. Okay, what about the other segments? In none other of the deletion, this segment is deleted, so they will all show the mutant phenotype. Sorry, they will all show the wild type phenotype. Okay, next, go to the next segment. So again, here is a deletion, so M. Okay, here is a deletion, so M. Okay, here plus plus plus. Now again deletion, so M, M, M. Here plus plus. Okay, this big segment in between, it is plus here, M, 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 and plus. In the next segment, it is plus, plus, and then M, M, M. So wherever deletion is there, the phenotype will be M. Okay, again plus, 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 M, M. Okay, now what you do, simply take this profile and match it with the table given here. Okay, so whatever gene will show that pattern means the gene is present in that segment. Okay, so see here, so first segment has M plus 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 plus. Is there any gene that is showing that pattern? M plus 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 plus, B is showing. Okay, that means this segment B is present. Next one, so like this you can do the entire analysis. Okay, so you just have to find, so this is the easiest way, so where you write the expected phenotype and then you correspond it or match it with the table given. Okay, so next one is saying mm plus plus plus. Okay, so mm plus 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 is shown by C, that means this second section C is present. Next is mmm plus plus. Okay, so mmm plus plus is shown by D, that means next is D. Next segment is showing plus MMM plus. Now here we do not have any gene out of this A, B, C, D, E, which is showing that pattern, which means none of them is present here. Okay. Go to the next column. It is saying plus plus MMM. So plus plus MMM is shown by E. So E will come here. And last one is plus 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 MM. So it is A which is showing that pattern. So A will come here. So now you have got the gene order. So what is the order will be of these mutations? B, C, D, E, A. So answer will be option 1. Okay. So again, for solving this question, the only thing you have to remember that in deletion mapping, the genes which are present on the deleted segment will have the mutant phenotype. ओके तो अगर आप इसको म्यूटेंट फिनोटाइप बोलना है तो वो बोलो, रिसेसिव फिनोटाइप बोलना है तो वो बोलो, ठीक है, बट दोनों चीजें सेम ही होगी, राइट नाउ। और बाकी जगहों पे क्या होगा, जो भी जीन डिलीटेड सेगमेंट में प्रेजेंट नहीं है उसमें कौन सा एक्सप्रेशन आएगा, वाइल्ड टाइप और द डोमिनेंट एक्सप्रेशन। ओके सो जो यहाँ पे डिलीशन का टेबल दिया हुआ है उसमें आप पहले लिख लो कि कैसा एक्सप्रेशन आप एक्सपेक्ट करते हो, राइट, एंड देन मैच इट अप विद द एक्सप्रेशन टेबल, और जो भी जीन वैसा एक्सप्रेशन दिखा रहा है जस्ट पुट इट देयर। ओके सो दिस इजीली you can solve this kind of deletion mapping questions, which are also have been asked multiple times in the CSIR exam. Okay, another question, let's move on. Now this is also linked with pedigree and molecular mapping or the molecular markers. So pedigree given below follows the inheritance pattern of a late onset after age of 30 years genetic disease that is 100% penetrant. Okay, so what is this disease? This disease is a late onset disease. Late onset means it will happen, but it will happen after age of 30. Okay, and but this disease is 100% penetrant. So we have already discussed penetrance in a previous video. So you know that penetrance means, if 100% penetrance means whoever has the positive genotype will also produce the phenotype. Okay, but that phenotype will come after at a later age. Okay, after the age of 30 years. Okay, there won't be anybody in the population who has the genotype but the phenotype is not produced. Okay, because this genetic disease is 100% penetrant. Now affected individuals are indicated by solid circle or solid square. So circle means female, square means male. RFLP analysis of DNA from each individual is shown in the pedigree. Okay, so here is the pedigree chart given and here is the RFLP pattern given. Which grandchildren, that is 3A to 3D, will be affected by the disease after attaining the age of 30? Okay, so basically it is saying that generation 1 and generation 2, they are all of their ages above 30 years. Okay, so that's why we know whether they are producing that particular genotype or not. Sorry, whether they are producing that particular disease or not. Okay, but for the third generation is still below 30 years. Okay, so we are not sure whether they will, like, whether they have the disorder or not. Okay, but we want to find it out before time. Okay, we don't want to wait till 30 years and then only check. We want to find it out before only whether these individuals are affected or not. So we are getting this information. Okay, so what we are doing, so here we are doing the molecular analysis. Okay, we have taken out the DNA. So even though the phenotype is not formed, but if the disease causing allele is there, okay, then it will form a particular pattern. That is what we want to understand. That is what we want to determine from this molecular marker mapping, to find out that if those individuals have a risk of developing the disease after the age of 30. Okay, so you have to find out that from 3A to 3B, who will be affected by the disease. Okay, so for this, what we have to analyze. So first we have to check the pattern. Okay, and then we have to check the the band pattern that we are getting for the individuals who are affected. So who are affected? Okay, so generation 1A is affected, 1C is affected, and 2B is affected. Okay, so who are they? I'll just mark them. 1A, 1C, and 2B. Where is 2B? 2B. Okay, so the columns where I have ticked, they are the one who are affected. Okay, so now we have to find a band pattern which is common to all three of them. Okay, so what do we see here? That these are the ones who have these two bands. Okay, and they have only these two bands, not the other one, because you see the not affected individuals like 1B or 2A. Okay, so for example, 2A has these two bands, but along with that it has this band also, and that is why 2A is not affected. Okay, the affected individuals are having only this lower two bands, right? So what does that mean? So from this third generation, you have to find out those individuals only who are having this lower two bands. Okay, so who will be that? 3A has it, but along with that 3A has this higher band also, so it will not be affected. 3C also have it, but 3C has this band also, so not affected. Okay, so only 3B and 3D are the one who have only those two lower bands, which is matching the profile of the affected individuals in the early generations, the higher generations. That means they are the one who will be affected. 3B and 3D only. All right. So again, analysis, and these are actually the practical application of the molecular markers. Okay, so molecular markers are routinely used in genetic disease diagnosis. Okay, by these molecular approaches, that even before the phenotype is produced, even before the symptom of the disease comes up, so the DNA, there is no information which is hidden from the DNA. Okay, so if we are able to analyze that, if we are able to collect that information from the DNA, so proper steps we can take, and the diagnosis of the disease can be done at an earlier stage. Okay, so these are the practical applications, and let me tell you that nowadays a lot of question comes from this molecular markers. Okay, this kind of analysis thing. Okay, and this is definitely one of the practical application. This is how a genetic counselor will work. Okay, or a person who is involved in doing the genetic disease diagnosis, this is how that technician, that expert genetic counselor that we call, will work. Okay, so if you are looking for a career as genetic counselor, definitely this is something that you should be clear with, and that is why nowadays in CSIR question paper, this kind of questions are very common. Okay, which makes you actually ready for doing a research in this field. Okay, so yes, we are approaching time, so we will solve one more question here, and before that, one more question, and then we will go to the Zoom class. So again, I request, please write down your doubts, come to the Zoom class, and we can have a one-on-one discussion. Yes, so this question, 3 somatic hybrid cell lines designated as X, Y, and Z have been scored for the presence or absence of chromosome 1 through chromosome 8, as well as for their ability to produce the hypothetical gene product A, B, C, and D, as shown in the following table. Which of the following option has most appropriately assigned chromosome for each of the given genes? Okay, now this is related to somatic hybridization. Somatic hybridization is also a technique which is used for physical mapping. Okay, so in somatic hybridization, basically we make hybrid of somatic cells. Okay, so for somatic hybridization for human genome, the physical mapping of human genome, the hybridization is done between the mouse cell and the human cell. Okay, now what it says what has been observed, that the somatic hybrid will retain all the chromosomes of mouse, but it will start slowly deleting or losing the chromosomes of the human genome. Okay. Ultimately, an equilibrium will be reached where all the mouse chromosomes will be retained, but only few human chromosomes will be retained. Okay. Now, what are the chromosomes from the human genome that are retained and how many of them are retained will differ from different cell lines. Okay. Now, this hybrid cell lines are further analyzed for the presence or absence of the different proteins. Okay. The simple correlation that is made is if the protein is present. Okay. Protein present means what? The gene coding for the protein is present. Now, gene ऐसे हवा में तो रह नहीं सकता, जीन ऐसे साइटोप्लाज्म में भी नहीं मूव कर सकता है, तो जीन कहां पे होगा? अगर जीन है means the chromosome which is carrying the gene will be present. Okay, so if a hybrid cell has the protein, means it will also have the chromosome that carries the gene for that protein. Okay, so this is the simple correlation that is made. Now, if you have a hybrid cell line that does not have the protein, okay, means it will also not have the chromosome that is coding, that is carrying the gene coding for that protein. Okay, so ये एक simple correlation है जो कि यहाँ पे लिया जाता है. Right. So which of the following option has most appropriately assigned chromosome for each of the given genes? Okay. So for that, see the gene products expressed. So let's see this column first. So the gene product expressed is for A is showing an expression of minus plus plus. Okay. तो क्या लगता है? जो chromosome होगा that is carrying the gene for this product A will also show the same type of expression, that is minus plus plus, right? तो कोई भी अगर क्रोमोसोम ये वाला एक्सप्रेशन दिखा रहा है, that means the gene for this product A is present on that chromosome. ठीक है, तो यहाँ पे 8 क्रोमोसोम का प्रोफाइल दिया हुआ है. प्लस मींस एब्सेंट, अगर खाली है मींस इट इज सॉरी द प्लस मींस प्रेजेंट, आई एम सॉरी, प्लस मींस प्रेजेंट. ओके, एंड अगर एम्प्टी है मींस एब्सेंट. ब्लैंक विल मीन एब्सेंट. ओके, सो माइनस प्लस प्लस कोई क्रोमोसोम है जो वो पैटर्न दिखा रहा है? यस, क्रोमोसोम नंबर 5. ओके, सो ए ही यही पैटर्न दिखा रहा है, माइनस प्लस प्लस, और क्रोमोसोम 5 also is absent in X but is present in Y and Z. Okay, that means A is present on chromosome number 5. Now let's see B. Okay, I'll just change the color. So B is plus minus plus, plus minus plus. Okay, so again, is there any chromosome which is showing this pattern? That is blank plus blank. It is chromosome number 6 that is showing the pattern, right? Which is sorry. I have done wrong. Okay, yes, I'm sorry. So what it is showing? Plus minus plus. Okay, so which is present in X, absent in Y, and present in Z. So it will be chromosome number 3. Present in X, absent in Y, present in Z. Okay, that means B is present on chromosome number 3. Next is C. Okay, now we will leave C. C is the tricky part. I will explain you. Now for D, okay, so D is present in all, means it should be present on the gene of it should be present on a chromosome which is present in all the 3 hybrid cell lines. Okay, so is there any chromosome like that? Yes, 1, right? That means D is present on 1. Now what about C? Okay, so from this pattern, C is absent in all the hybrid cell lines. Okay, now chromosome number 8 is also absent in all the hybrid cell lines. Okay, but along with chromosome number 8, we are talking about human chromosomes. So there are other chromosomes also till chromosome 22. Okay, also the sex chromosomes, right? 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, along with the 22 plus the sex chromosomes. They are also absent in these hybrid cell lines. So if this gene is absent, okay, this gene product C is absent in all the 3 cell lines. So that gene can be present on 8, but at the same time, it can be present on any of the chromosome which is absent in the hybrid cell lines. Okay, so we cannot say that C is present on 8. This is the tricky part, because when you see the options, okay, so option, so what we have, A on 5, okay, so A on 5, B on 3, and D on 1. Okay, so that is given. So this 2 and 3 options you can eliminate. Okay, so A on 5, B on 3, and D on 1 is given in both option 1 and 4. What is different here? Option 1 is saying that C is on chromosome 8. Option number 4 is not talking anything about gene C. Okay, so that only is correct. Option 4 is correct, because even though 8 is absent, C is absent, but we cannot do the correlation here, because along with chromosome number 8, all other chromosomes till chromosome number 22 plus the sex chromosomes are also absent here. So this C gene can be present on any of those absent chromosomes. Okay, so that is something that we cannot conclude here appropriately, right? That's why the answer is not option 1, but it is option 4. Okay, so that was the trick part here. Okay, many of you might have selected option 1, right? And this somatic hybridization questions are very, very easy. So if there is only one gene that you have to place on the particular chromosome, so usually it is asked in part B only. Here there were 3 genes that you had to place, and there's a trick part also involved, and that is why it was put in part C. All right. Okay, so you have to be careful. You have to choose the correct option. You have to analyze the questions properly. Okay. All right, guys. So we finished 5 questions and we are passed with the time as well. So we'll stop the live session here. I request the team to put up the link for the Zoom class along with the link for the freebies. So from those links, you can download the freebies that will help you in your CSIR preparation. Okay. Along with that, please team load the class, the Zoom class link as well. The Zoom class session will start soon. I'll be waiting there for all of you to come and clear your doubts. If you guys do not have any doubt, we can discuss some extra question as well. Thank you everyone for the Zooms, for this live session. Bye. See you soon in the Zoom class.